Okay, welcome everyone. Thank you for coming to this wonderful event, the opening day of uh, the official opening day for the Institute for Matter and Systems. But more importantly, we're here today also for the Oliver Brand Lecture. So most of you know Oliver. He was a pioneer in MEMS. I think most importantly, he was fantastic at bringing faculty together and was a colleague and friend of mine. And we really miss him. And we really hope we've set up a wonderful fellowship in Oliver Brand's name. And you can give as little or as much as you want, but I think it's a great way to support Oliver's legacy. Also, we have here, if you want to learn more about IMS, our LinkedIn page, Twitter page, newsletter, please sign up. My name is Eric Vogel, and is it working? All right. And I have the pleasure of directing this new institute, the Institute for Matter and Systems, that came about by bringing together the Institute for Electronics and Nanotechnology and the Institute for Materials. I'm just going to take a few minutes to talk about the Institute for those of you that don't know. So one way to think about where IMS falls into technology, innovation, and science is through the lens of national priorities. So if you look in 2000, we had the National Nanotechnology Initiative that we've been heavily involved with through NNCI. In 2011, my predecessor, David McDowell, was heavily involved in a lot of folks at Georgia Tech with the Materials Genome Initiative. And of course, in 2022, the CHIPS and Science Act. There are certainly differences in all of these national priorities, but one of the common themes is the idea that technological transformation many times happens when you bring together the fundamentals of matter, of science, through devices, process components, but then you really link that to final systems and to society and economics. And this is really the Institute for Matter and Systems is the core of what we do is related to materials, processes, and devices, but we're really trying to link people from fundamental science through materials, measurements, modeling, software, systems, economics, public policy. That's really what we're trying to do in the Institute for Matter and Systems. And we do that in 4 ways. These are really the building blocks of what we do. So in the center of this circle, I'm going to talk to each of these, but we have research and innovation. So we support research initiatives and centers. We have fantastic fabrication and characterization facilities. We offer education and outreach programs, interdisciplinary programs that faculty can tap into. And we do a lot with strategic engagement with external stakeholders. I'm just going to talk briefly about each of these. So in terms of interdisciplinary research, we support interdisciplinary team formation across Georgia Tech, sort of from, again, the College of Engineering, Science, Computing, Business, Public Policy. We provide support for proposal development, communications, and events. And we do this through research initiatives, programs, and centers. So research initiatives are initial small amount of funding to help start bringing faculty together. If those start to become successful, we provide some additional support for our programs and our centers. And we currently have 7 interdisciplinary research centers and 16 of these programs and initiatives with over 100 faculty that are engaged in these research activities. And if you're a professor here on an annual basis, we have a call for these research initiatives. So you should be looking out for that call coming in the spring. And we do research right now. We're grouping into 4 main categories. So we have a lot of work going on, obviously, with microelectronics. Secondly, what we're calling built environment technologies. So think automotive, infrastructure, energy, human-centric technologies, and then cross-cutting. So looking into how innovation happens, economics, as well as characterization, things that really cross all of these different technology areas. And so I'm just going to give you an example of 3 of our centers. So the Center for Organic Photonics and Electronics, it's led by Jason Azoulay and Juan Pablo Correa-Bena, really look at organic polymeric materials for everything from optical signal processing, solar energy, sensor development, flexible transistors, and have been really successful. We have a whole cohort of faculty over the last several years that we've brought that Georgia Tech has brought together. So that's one example for microelectronics. A second example, the Georgia Tech Advanced Battery Center. So this is led by, it's first in collaboration with our Strategic Energy Institute. It's led by Matt McDowell and Gleb Bushin. They're bringing faculty together to look at all kinds of different battery technologies, lithium-ion, solid state. They're also in the process of setting up a translation facility where if you have a new material that you're looking at for an anode of a battery, we're going to have a translation facility that makes a reasonable number of pouch cells so that you can really see how a final battery works how your material works sort of in a final battery. The third center I'll highlight is led by Hong Yao. It's the Wearable Intelligent Systems and Healthcare Center. Again, very interdisciplinary. So everything from fundamental materials, understanding how sensors interact with the human body, out to working with medical professionals and doctors and doing measurements of actual people. And so this just gives you an example of the kinds of teams we're trying to bring together, really trying to cross Georgia Tech, really break down silos, break down the silos that many times occur in schools. The second thing that we do, of course, is we offer shared research facilities. So our clean room and characterization facilities. So we have almost 40,000 square feet of space. These are with 30 staff that can help you with your research, that train users, as well as do projects, over 250 instruments that supports the research of over 300 principal investigators and 1,000 unique users. These facilities are also used by academic classes. So we have almost 14 different academic classes from across Georgia Tech that come in for IC fabrication and for various aspects of materials characterization. A number that I like to give internally is if you look at the contract, the awards that are used to pay the bills for our facilities, the total funded award amount of those contracts is almost $500 million. And so almost a third of all Georgia Tech academic side research that is pursued here uses our facilities. So we have a huge impact on all of Georgia Tech research. I should also mention that we're the coordinating office for the National Science Foundation Nanotechnology coordinating infrastructure. And so David Gottfried is leading that effort, and we're coordinating 16 university sites across the country that also offer these clean room and characterization facilities. The third thing, we offer a targeted menu of education and workforce development programs. These are really interdisciplinary programs. And so again, if you are a faculty member and you are looking for ways to engage and to help with broader impacts for your research, we offer a menu, and you can talk to Micheal and he'd be happy to have you involved in our education and workforce development programs. So we lead a research experience for teachers program, an REU program. We collaborate with Micron to offer a summer CHIPS camp to K through 12 students. And then finally, we work, and Judd Reedy is leading this part, we work to have strategic relationships with a lot of external companies, government agencies, and foundations. So we have fellowship programs. BASF, for example, helps support PhD fellowships. We coordinate internships with Sandia National Labs. We work with our Office of Corporate Engagement to make sure we know what kind of technologies are available at Georgia Tech in the area of materials and nanotechnology and devices for licensing. And then we do these kinds of programs. And we have many symposia, the Oliver Brand Memorial Lecture, of course, the Systems Matter Seminar, and a variety of workshops that we offer. So if you're interested in getting involved in workforce development and education, please let Micheal know, and external engagement, please let them know. So I did that very quickly, but we're really happy that we're here. I want to thank everyone that has helped to put this together, Rebecca Grieco, Amelia and Cecilia, and many others that helped put this event together. So let's please give them a I want to thank the rest of the employees of IMS, the 60 to 70 of you that work in our facilities, handle finance and proposals and communications and everything else for what we do. So thank you for making this a really great day. And it's been a joy working with you over the last year. And so with that, what I'm going to do is turn it over to our Deputy Director, who's responsible for research and innovation, Mike Filler, who will introduce today's speaker.
Thank you, Eric. Good afternoon, everyone. As Eric said, my name is Mike Filler. I'm the Deputy Director of IMS. I'm also a professor in the School of Chemical and Biomolecular Engineering here at Georgia Tech. It is my honor to welcome you to the second Oliver Brand Memorial Lectureship on Electronics and Nanotechnology. Today we gather in the Marcus Nanotechnology building to remember a remarkable individual, Professor Oliver Brand. His contributions to Georgia Tech and to the field of electronics and nanotechnology. We're honored by the presence of Oliver's family, his wife Claudia and daughter Marina. Your presence here reinforces the profound impact that Oliver had, not only as a scholar and a colleague, but as a cherished husband and father. As we honor Oliver's memory, it's my privilege to share more about his academic journey and the life he led with such distinction. So after completing his undergraduate degree in physics at the Technical University of Karlsruhe in Germany in 1990, Oliver pursued a PhD at ETH Zurich in Switzerland, which culminated in 1994. He studied electromechanical systems, or MEMS, during his doctoral studies, which laid the foundation for his independent career. He joined the Georgia Tech faculty in 2003. His contributions were vast and varied. His work, especially in MEMS and CMOS-based microsensors, is widely respected with over 190 publications to his name. Oliver served as the executive director of the Institute for Electronics and Nanotechnology, which is the predecessor of the Institute for Matter and Systems. And prior to merging with the Institute for Materials to become IMS, in his role as IEN director, he united Georgia Tech researchers and technical staff to support infrastructure for and advance interdisciplinary activities in the areas of electronics and nanotechnology. IEN boasted and still boasts nearly 30,000 square feet of cleanroom space, hundreds of pieces of equipment for cutting-edge research, and IEN's over 200 faculty member affiliates and nearly 50 full-time staff at that time made it one of the largest research institutes at Georgia Tech. Oliver also directed the coordinating office of the National Nanotechnology Coordinated Infrastructure, the NNCI. NNCI is a network funded by the National Science Foundation that provides researchers from academia, small and large companies, and government with access to leading-edge nanofabrication facilities. The NNCI's 16 sites encompass a wide array of capabilities and expertise in nanoscale science, engineering, and technology, supporting and advancing research and innovation in the field. Oliver also served as the director of our local NNCI site, the Southeastern Nanotechnology Infrastructure Corridor, or CNIC. Beyond his academic success, Oliver was a mentor, a colleague, and a friend. He was known for his sharp mind, his gentle style, and unwavering support for those around him. His ability to foster collaboration has left an indelible mark on the community near and far. I'm particularly pleased to note the portrait of Oliver that you walked past on your way into this room. It stands as a symbol of his enduring legacy. It's a fitting tribute to a person who dedicated their life to advancing our field through scientific excellence and community building. And today we are privileged to hear from Professor Micheal Strano, the Carbon P. Dubs Professor of Chemical Engineering at the Massachusetts Institute of Technology. With a distinguished academic background that includes a Bachelor of Science from Polytechnic University in Brooklyn, a PhD from the University of Delaware, and a postdoc at Rice University under the guidance of the late Nobel laureate Richard Smalley. Professor Strano has made significant contributions to the field of electronics and nanotechnology. His research is widely recognized for its impact on the surface chemistry of low-dimensional systems, nanoelectronics, nanoparticle separations, and more. His many accolades include the 2005 Presidential Early Career Award for Scientists and Engineers, the 2006 Beckman Young Investigator Award, and his election to the National Academy of Engineering 2017. Today, he will share a talk titled Nanoelectronics Grafted onto and within Colloids for Colloidal State Machines with Micro-Robotics, and it is one I eagerly await. Following today's lecture, I invite you all to join us for a reception in the atrium and the rest of the day's events. Thank you for being here to commemorate Oliver. Let us carry forward his legacy of curiosity, collaboration, and compassion, inspiring future generations in the field he so dearly loved. Now, please join me in welcoming Professor Strano.
Okay, there we go. Okay, great, wonderful. It's my pleasure to be here today. It's really my honor, and I've had a wonderful morning, and it's been great to be here on campus. I have many collaborators and friends, and I wanted to start off just saying my own few words about Oliver. I didn't have the pleasure to meet and interact with Oliver, but all of us in the field knew about the work here at Georgia Tech, and anybody that has read the New York Times knew the impact that Oliver had, particularly during the pandemic. And I think I can't say anything better than what Oliver's colleagues have already said. They said he was a pioneer and described him as transdisciplinary, and has had just an enormous impact. Few can match his the magnitude of his influence on campus. And during the pandemic, I think everybody that worked on sensors also turned their attention to the virus. It was the one way that we could receive permission to work in our lab. So we also were working on a COVID-19 sensor. And but Oliver and his team really did it, and they accomplished it, and it had a huge impact. And even leaders at the NIH described the team as absolutely heroic. And so it's tall shoes to fill, and it's my honor to be here. And it would be fantastic if this lectureship also invited speakers that were transdisciplinary. So I hope that's the theme. And I will introduce myself. I'm an unlikely academic to talk about robotics. I'm a chemical engineer by training. I always like to start off with advertising for postdoctoral associates. But here's the team. This topic that I'm going to discuss has really been about a decade worth of very slow, certainly by Oliver's standards, very slow progress. And so I have to advertise some of the early pioneers. This is a picture from the summer of 2018, and then this is a recent group picture. But my group in general, what I work on, I work on engineering at the nanometer scale, and that's also Professor Brand as well. I'm interested broadly in how matter changes when you pattern it at the nanometer scale. So these are gold atoms, and they're chemically reactive at the nanometer scale. But in the bulk scale, we make jewelry out of it. It's not reactive. So the gold in my ring is inert, but at the nanometer scale, it can catalyze chemical reactions. This is a single-walled carbon nanotube. It's maybe one of the smallest wires, but you can pass electricity through it. It's very different from the wires that are carrying electricity in this room. And mass transport also, pores that can translate DNA through them. The rules of mass transport change at the nanometer scale. And so the fundamental logic of my group, I look for these unique properties, and I try to solve longstanding problems with these changes in reactivity, the way heat and electrons move, and mass transport. So I'm going to talk about micro-robotics, and Georgia Tech has been a leader in robotics. The students at MIT were worried that you would think that that's the only thing that I work in. I want to give just a little bit of a whirlwind tour of some other areas that I work in, and one is the area of sensors. So I work on single-walled carbon nanotubes that fluoresce, and we use them as biosensors. And in fact, I was pleased to see that Georgia Tech, you hired a brilliant new faculty member in chemistry, Mi-Jin Kim, and she works in this area and is doing fantastic, amazing things. But the carbon nanotube will fluoresce in the near infrared, and then what we do is we functionalize it with a variety of polymers, and we try to change its. This is supposed to be its fluorescence. We change the intensity of its fluorescence or its wavelength as molecules bind. And there are lots of applications of this. We've used it to place small sensors inside living plants. This is inside of a plant leaf, and you can see the internal chemical signaling in real time in your living plant. So you may not know, but plants actually talk to themselves inside, and you can actually see those chemical signals. We've made tools that can detect cancer therapeutics inside mammals, and we've been tagging marine animals for a field called biologging. So I won't talk about this today, but it's been a fun area to work in. And now Georgia Tech has a fairly large research footprint. I started this area called plant nanobionics. This is where you take nanoparticles, you put them into living plants and try to give them new functions. One function we've been giving them is to turn them into light sources. So this is a light emitting plant. It uses 3 kinds of nanoparticles. And the idea is it can intercept some of the chemical energy that the plant produces, and you can make a light. And it's part of an effort that I've been working on at MIT to replace the things we make out of circuit boards and plastic, to try to replace them with things that are renewable. And so you can check out our work on that as well. We've since turned our attention to sensors for the plant itself for agriculture, monitoring things like drought. And we've even made an explosive sensing plant. This actually went viral. It was on the Daily Show. I'm not sure why, but it's a living spinach plant, and it can detect explosives in groundwater and then send an infrared signal to a user's cell phone. And I think Oliver maybe would have appreciated that, and maybe would have found a better use for it than what we have. I also, not to belabor it, but I also work in the area of understanding the thermodynamics and transport of fluids inside of those carbon nanotubes that I showed you. So we put fluids inside of the nanotube. This is a spectrum of what happens to a vibrational mode as we induce a phase change in the fluid. And this is an area of great interest to the field. We're working on a confined fluid equation of state to describe what happens to a fluid that's essentially just a wire. And just as an advertisement, I started this Center for Enhanced Nanofluidic Transport, and it's a Department of Energy sponsored center with 16 faculty around the nation, and we're trying to study problems like this. What happens to the thermodynamics of what we call ultra confined fluids? Last but not least, we'll get to robotics soon. Is we've been working on 2-dimensional polymers in the form of this 2-dimensional polyaramids. This is, you can think of this as 2-dimensional Kevlar. And we figured out how to polymerize them. We spin coat them, and they form these interesting layers. They're exceptionally strong, and they have properties that are somewhere between a polymer and inorganic materials like graphene. And so right now, we've been studying their ability to trap gases. These are experimental bulges. We can fill them with gases, and they're completely impermeable, even at a very small nanometer scale level. They're the most impermeable polymer on record to date, and this is an AFM of that bulge. So that gives some of the scope of what we work on. I'd like to think that I am transdisciplinary, and so maybe it does fit within the lectureship. But what I came here to talk about is an area still in progress. It's we call it colloidal robotics, and I'll explain why. And the students have packed a lot of projects in here. They really want to show you quite a bit of what we've done. I chose this area because it's solidly electrical engineering. I'm a chemical engineer, but I thought it would be appropriate and would and would fit. But before I start on our evolution here, I wanted to just give a background. Why do we care about? Is it crazy to graft electronics onto colloids? Aren't colloids mayonnaise and milk and so forth? Well, a colloid is a particle that dispersed in a fluid that doesn't settle, and so they can range in size from hundreds of microns to down to 1 micron or nanometer. If you spray droplets into the air, you have a colloid. There are solids. I mentioned that they're in milk. Colloidal electronics tries to graft electronics onto these particles, and it's something that nanotechnology can do quite well. Now, why would you want to do this? Well, the idea is to bring electronics to environments that are inaccessible. So the idea is to make maybe an electronic device or robot that's the size of a living cell, and be able to access spaces. Like a big dream of the field is to be able to put them into the bloodstream. You could also think about small particles that are functional devices that you could spray on. They could self-assemble, and you could make really sophisticated electronics really just by spraying instead of a laborious clean room process. And I know Georgia Tech, you're a leader at those processes, and they work well. But could we think about how to assemble sophisticated electronic devices built up as little functional cells? It's not a crazy idea. It's how your body functions. You're made up of little semi-autonomous cells, very sophisticated in nature, and they're pieced together. We humans do not build materials like that in a cellular way, but maybe we will. But in order to do that, we need to think about colloidal electronics. And so it's an ability. Could we use microfluidic platforms to then build cells that we can then assemble? And we live in an age of really tissue cellular printing, so it's not too far fetched. But there's a lot of basic engineering that has to be solved. We're actually not ready to do this year or next year. There's some basic engineering that has to be solved. Actually, while Oliver was saving us from the virus, I was writing this review. Okay, it took 3 years to do this perspective in Nature Materials. It's called Colloidal Robotics. I'm very pleased that it was published. 2 of my Georgia Tech colleagues here helped me. Dana Randall, brilliant in computer science, and of course Daniel Goldman is a real roboticist. And they gave excellent perspective on this nascent field that we call colloidal robotics. So I'm very, very pleased here. And in fact, Dan's work, Dan has been a pioneer here at Georgia Tech in making exceedingly simple robots. They just really sort of flip back and forth, but you put them together and then collectively they can do an astounding thing. They can move and locomote. And this has been a theme of the field of robotics. There are these kilobots from Rubinstein and particle bots from our own Daniela Russ at MIT. And when my and an example, of course, is in cartoons. When my kids saw that this was in my talk, they became very excited. They say, "When are you going to make this?" I said, "Maybe not too soon." Okay. But that's the dream. Small robots collecting together to do a collective task. Micro-robotics and colloidal robotics can scale enormously, and to utilize them, we need to tap this research. In the small robotics field, we call them, if you Google micro-robots, you'll see very sophisticated things like this, but they're not quite micron scale. So what I'm going to talk about today is micron scale. These still are pioneering. These are origami robots from my colleague Daniela Russ at MIT. Notice it moves around, and this is Kip Parker at Harvard, who used cardiac myocytes blended into a tissue, and you can make this movable stingray. Now there's a subtle thing here, and that's what you'll notice in both of these cases. Both the energy and the computation come from outside the system. And so I actually wrote a perspective in Nature that framed this. These we call marionettes, and they're very important. For one, they pave the way to what could be done if we could get past computational and energy constraints. but two, there are many applications like surgery or exploring the environment where it's okay to have a marionette. But today I'm going to focus on a much harder problem of, can we make a device that really is fully autonomous, not really tethered to its environment. It has its own energy and its own computation, and then they become less impressive than this. There's more work that needs to be done. But that is where the field wants to go. There's this, can we shrink a device down and put it inside the body? It's starting to become a reality in that myself, I'll talk about today, my colleagues at MIT, this is a system from Cornell. We're now at the point where we actually can make a cellular robot. This is one that is, I would say, is autonomous. It's getting energy from a laser pointer going to 2 different photovoltaic cells. I would say this is autonomous. Still getting energy from outside the system, but maybe it doesn't need the computation. But we're building devices like this. We're at the stage where we're learning how to build devices like this. And so that's what we want. We want particles. We want them to be able to actuate, which is very hard, sense things. We want communication. We want energy. We want all of that into a particle. And we want it for all these reasons, to bring electronics to inaccessible areas. Spray-on fabrication. Can we make, is this the future of robotics? Robotics made in this cellular way. So that is the vision. This is, almost everybody in the field shows this picture. It's from a 1966 movie called Fantastic Voyage, and the scientists shrink themselves down into the submarine. It is it's aspirational. Okay, we'll put it that way. Not the shrinking down humans, but putting something into the bloodstream. And it's something to, it's a goal, a long-term goal, but it gets you thinking about, how do I do engineering of something that can access that space? You encounter a lot of problems that are useful to solve in general. And I actually wrote a book with Sean Walsh at the Pentagon, and it really frames materials problems in this area as a manufacturing one, and hopefully one that colleagues want to solve. So, what scale do we mean? There's pioneering work with, we saw a reference to Aberson's work here on campus. These ingestible cameras. These are millimeter scale. They're called micro systems, but they're centimeter in scale, and they're already having an impact for imaging the GI tract. We want to get down to red blood cells, so that's about 7 microns. So we really want to assemble in this area, and it's a huge leap, and I'll explain why. If we go, it's one of the most important things to point out about this area is the discontinuity in energy. It's not intuitive. You really have to study it to understand it. This is in electronics. There's this famous log scale, length scale. We even saw one from Eric's presentation. Down here at 1 nanometer and 10 nanometers, that's the scale of maybe macromolecules. So here, actually, the system is in a thermal temperature bath. It's literally swimming in energy. So actually, there's a whole field. There was a Nobel Prize in making molecular machines, but they have access to really all the energy in the world, okay? If you look at where electrical engineering is, we're not the first to think about small electronics. There's this idea of smart dust in the 90s and micro robots that are millimeter scale. Well, these are these devices. They are promising, but I think the dream has always been to get to this really dust. Dust is microns, so really in this space. So when you start to run numbers, what you realize is for most of these devices, the energy demand that they require scales with area. So for electronics, the bigger I make an electronic chip, it actually goes as the area. That's the way we print electronics, right? Same thing with drag. Drag on a particle is an area. It goes as L squared. Oh, yeah. Whoops. Sorry about that. It goes as L squared. Let's see if we can go here. But energy supply goes as L cubed. So if I take your laptop battery and I shrink it down to 1 centimeter by 1 centimeter, I don't get any more energy. I get slightly less. So energy scales with L cubed. demand scales with L squared. And out here, you're in the energy-rich environment. Okay, this is where children's toys are, and it's where you have plenty of energy. This scaling suggests that after this this barrier here, that might be my phone. I'm really sorry. I'm not, that is, I'm going to say, I'm going to shut this off. Okay. Anyway, I was making the, okay, that'll teach me really. Yeah. So anyway, long story on this side, there's no fun. You don't have any energy to do even the basic things that you want to do. So you really have to be miserly here. And I think this divide is stark. So if you Google a lot of fancy devices, you'll see videos here. You'll see much fewer, maybe even none, very basic things here. But if we want to get to the colloidal electronics vision, we need engineering here, and there are lots of unsolved problems. Okay, it's not an area that I recommend for an assistant professor, or it's these are tricky problems. So in that context, I'm going to talk about a few projects that we've done in this space. The first is aerosolizable electronics. Can you make a particle, aerosolize it, and have it do some functions? Now, why would I want to aerosolize it? Well, that gets at getting electronics into spaces where you wouldn't normally want them, and I'll explain that. Where you normally, I'm sorry, I did not mean to say normally want them. I mean where they would not normally be accessible. So we made these cell-sized robots. They were our first, and we used a standard electrical engineering approach, and I'm hoping that the audience here would appreciate that. These diagrams are very similar to what you do in the microelectronics field. So I'll explain this. This is a copy of our chip. It's on SU8, and we lift it off. It becomes a particle. It's in solution, and I think it's the first fully intact self-powered nanoelectronic circuit. And I'll explain what we did here. Is so first you can clean room fabricate this, but then you lift it off, and they're particles in solution, and they're very stable. And we've learned the hard way. You actually can't do this with conventional silicon electronics. You actually have to use 2D materials like MOS2 and graphene. And so what this particle does is it has a photodiode built from an MOS2 flake junctioned with tungsten disolenoid, actually not a flake, CVD heterojunction. A separate flake actually gives you a chem resistor, and then with contacts, a mem resistor. And you contact these as a circuit. It's not, I mean, it doesn't seem that impressive, but light can actually be powered, light can power the photodiode, and then it can write to the mem resistor if the chem resistor is triggered, okay? This can all be done in a cell-size footprint, and it does this logic. So it can, if it detects light and it detects a chemical, it can then write to this 1-bit memory, okay? And for 2018, it was state of the art at the time. So light comes in, powers the circuit, and then if there's an analyte, it will allow the circuit to write to the memristor. This memristor is a 2-terminal memory, and these are the electrical engineering symbols. So we built a test cell. We could spray these, and if they encountered soot, the MOS2 sensor would would change the resistance of the memristor, and then they would write to the device. So we would inject these, and you could actually harvest. We would harvest these on microscope slides to demonstrate. This is the memristor conductance. This is the chem resistor conductance. And you can see here that if you have both soot and light, there are devices that would trigger, and they would write to the memristor. And at the time, we were collecting them, and we could query the memristor, and we could see that they stored that information. So we learned a lot from the building of this device. We also we showed that they could also detect ammonia, and that brought us to an application. We think that probes like these could flow in industrial pipes, and they can look for leaks and environmental hazards. They work perfectly flowing down. They can flow through miles and miles of pipe, and they have this ability to detect to remember, to store that information. And so they're a small sensor that moves towards this vision. We've been thinking a lot about the bioethical considerations from day one. So these particles are so small, I could spread them on this desk and you wouldn't be able to detect them. So we're trying to build in safeguards from day one. So you can't see them by eye, but you can put these retroreflectors on them on both sides. And then actually with just a laser pointer like the one in my hand, you could scan and you could you could easily, even on this desk, I could scan and see the retroreflected light. So it's a way to track them. And I think we need to build those kind of safeguards in from day one. We're really responsible for every single one of these particles that we make. So that's aerosolizable electronics. That got us into new ways to fab. And so this is actually a new way to fabricate things that are starting to look like cells. And again, we wanted to really move out of the clean room. This takes 18 steps, the failure of which makes you have to start back to step one. Anybody that's worked in a clean room, maybe there's students here, you've got these 18 steps. If one goes wrong, you have to start over. So we're looking for shortcuts that we can do outside the clean room. And there's pioneering work here at Georgia Tech on additive manufacturing and so forth. We need more of that. But we found a technique based on fracture. If I throw a baseball at the glass here, I'll get a spider fracture. We don't make things by fracture. There's nothing. Do not make your electronic devices here by breaking and fracturing things. It's a bad idea. Fracture is random, but actually it can be controlled. So the person who figured this out was Griffith. Actually, if you calculate the energy to break glass, the chemical bonds are so strong, you'd never be able to break it. We should be able to park cars on top of glass panes, but that's not true. Why does glass break? Glass breaks, actually, Griffith figured it out, that glass has stable micro cracks in it. If you look at the energy, if you calculate the strain energy to break, it's huge, right? It should be quite strong. But as a crack propagates, you gain surface energy. Griffith added these together and you get a characteristic length called the Griffith length. So materials that are brittle have a Griffith length. Cracks below that length are stable. So you see glass, but actually the glass has little tiny micro cracks and they're stable. When you throw the baseball at them, what happens is you increase the strain field and the strain field changes the Griffith length. And this is all, you can calculate this. And we're not the first to realize this. There's some work by Irvine and coworkers. They, at the macro scale, 5 centimeter scale, they put PDMS beads, and you can actually put, if you put a brittle film on top, you can actually guide a crack. This is guiding a crack along these. So we decided to see if we could get this to work at the nanometer scale. Can use a strain field. And so that led us to invent this technique. So you could take a 2-dimensional material like graphene, and infinitely molecular thin, okay, and you can print any combination of things you would like inside your cells. So these beads will eventually become the organelles inside your cell. And what you can do is, if you start off by doing that on a 2D film 1, you do a 2D film 2, you make a sandwich, and what you can do is you make these posts. If you apply liquid, you'll shear them and you'll get the system to fracture just around these beads. So it's a way of making a cell type particle, and you can do semiconducting materials like MOS2, you can do graphene, you can do insulating like HBN. And so the idea is if you apply a solution here, now you'll propagate cracks, but when they get to the high strain field, they will crack. You can see here, you can just lift off. And what you have here now is a sandwich. You actually have control of surface A, control of surface B, and you're able to print whatever kind of organelles inside. So these things have the shape, form factor, and size of living cells. And so we think it's an important tool for this colloidal electronics. And if one of the things we did, well, but before I show you some applications, it's noteworthy that in solution, they actually form the same kind of toroidal shapes that a red blood cell forms. And in fact, you can scale them down, and you can study their flow. And one of the things that we did, I'm going to skip over this, but to get more to the electrical engineering, is we made MEM resistor arrays out of them. So this is something that we've been interested in. You can use what's called black phosphorus. You can put that inside the particle, and then that makes an array of what are called MEM resistors. And this is for the electrical engineers. This is the characteristic of a MEM resistor. This is graphene polystyrene graphene. This is graphene polystyrene black phosphorus. And you see you make a much better MEM resistor. So this is a MEM resistor is a device you can apply a forward voltage. You'll change the resistance to where if you sweep back, it'll remember you've stored 1 bit of memory here. Then you can go backwards and you can erase it. So it's a random access 0-bit memory. In an array, you can store multiple bits. So this is what we've done here is you can with a probe you can write to these. And so we wrote MIT here, and I'm very glad that we weren't at Georgia Tech for this, because it would have taken a lot longer to spell out Georgia Tech. But you got MIT here, where you can see that this particle now. One thing you can do is you can put very small bit arrays inside the particle, and so if so that's a technique that we've been using, but the elephant in the room is really energy, and so we after the pandemic we started an effort to really look at, can we make a battery? Can we at least make the best battery for cellular electronics? These cells need a mitochondria, okay? We looked around, and we first started working with lithium. So here's a smart dust to give you a sense of scale. Smart dust is about a microliter, and that sounds small, but for cell size, you need a picoliter. We need a picoliter battery. And if you want to, the students tried to show this here. Yeah, so that small dot is what we're trying to do. That is, so it's very, very different. A microliter is not the same as a picoliter, okay? In terms of, so we wanted something that was compatible with conventional lithography and something that could scale. And we moved away from lithium, and we went to zinc air. And there's lots of advantages here. So zinc air is, and it actually uses water air sensitivity to actually power. So it's not deactivated by air and water. It's actually powered by it. But it also allows us to start off with a charge state. We don't have to charge them, and it's and you actually can do very good photolithography with zinc. So I hope the electrical engineers would agree we've made a good choice. But I know there's other choices here. But so this is our zinc air battery, also on the SU8 substrate, and we're also using the Colloidal electronics platform for it. But you can see here, so we start off with an SU8 base, a platinum cathode, we put a titanium adhesion layer, and then you can put the zinc anode on, and you can power real loads with this. So you can, this is what the battery looks like. You can shrink it down to cell size. And we did a lot of just basic battery characterization. You can release these into solution. So here they are. These are cell size batteries in solution. They float around in solution. Depending on the buffer that they're in, they'll discharge and they can make a zinc phosphate. I'll explain that. But otherwise, they're free to power a wide variety of loads. And we did that in this paper that we published this year. So one of the first things we did is we studied how the batteries work in different buffers. And we found that if they're in PBS, this phosphate buffered saline, which is a close mimic to biological fluids, the tear fluid in your eye, many areas of the body, you actually get much power out. You get a lot of power out. And the reason for that is you end up making a zinc phosphate. And so there's a particular advantage in doing this chemistry. And we thought that was exciting because actually this is chemistry that can occur inside your body. So this is a step towards bringing these devices into the body. Zinc is an essential nutrient, and so it is. It should be, just on the face of it, it should be more or less biocompatible, absent the reactions that can be catalyzed with the battery. So we looked sort of standard battery characterization. We looked at discharge curves. We get an open circuit voltage of about 1.22 volts, and then as you draw current, this makes a battery power curve, and so this tells you how what kind of loads you can attach. It turns out that it's of the right characteristics for powering things like sensors. You can do chemistry, you can do a little bit of propulsion and things like that, but there's not a huge amount of energy, but that's baked into the cake for this kind of problem. And these are the galvostatic curves. If you want to see here, this is for the battery people. It's enough. You can get stable voltage out until the battery depletes, and of order an hour, which is what we targeted, at least for this size device. And so the key here is that these characteristics are enough to power some of the functions that we wanted to do inside the for our battery applications. And it turns out, I'll just turn to this graph. If you look at, people have worked on microliter batteries, but actually there's very little work in the picoliter. Actually, this is, we're convinced this really is the best that you can do in a cell-sized battery, a picoliter battery. Okay, and so we think we got all the literature here. It's basically, this is where the MIT battery is, and so maybe it's the best that we can do for now, and it has all of these advantages. So what are we doing with it? Well, we're powering things like, this is the Cornell team. They made this walker. This is called a bimorph actuator. So the battery can power this actuator. It's not a huge amount of force, but we're starting to assemble. What I'm trying to show you is we're starting to assemble the robotic pieces in order to get things like this to work. And this just shows a little more of the data, switching back and forth. So I showed you this device, which had the laser pointer. In theory, if you put a battery onto it, you could have the thing walk, and it would be autonomous if there's water and oxygen around. Okay, great. So I have a little bit of time. I wanted to, because this is an electrical engineering audience, I wanted to present what I hope is an interesting idea of how we can use MEM resistors in this space. So I'm a chemical engineer. This is my first attempt at circuit design, and so maybe it'll teach me to leave it to the experts like Georgia Tech. But I'll explain to you what we. So I mentioned this MEM resistor, which I'm sure people in the room know what it is. I'm fascinated by it. 2-terminal memory. This is a MEM resistor, and the student. So this is what I wrote into one of these cells in this MIT. The students made, so if you see, I'm trying to show you that if you apply a voltage here with an array of resistors, what'll happen is the first MEM resistor will write. If it writes on, so current can flow, what you'll get is a succession. They're trying to show it here. Do you see the fade? It'll just keep writing to MEM resistors, right? And actually, we have figured out that that is a way of doing a very cheap onboard timer. That actually, this thing can keep time. And so I want you to imagine that you have a cell. Well, no, you don't have to imagine. This cell is an array of MEM resistors, right? So we started doing some calculations. And the calculations, you can simulate these easily. There's just a simple differential equation which describes the on-off boundary. It should be proportional to the current. This is all, I think, pretty well known in electrical engineering. It scales with the material. We, in theory, should be able to tune this time constant. But what we figured out in this paper is you can use an array of MEM resistors like this to keep time. And actually, it's arbitrary time. You don't have the frequency to double. And so we think this array can keep track of time. This a orthogonal array, if these are chemi resistors, I'm sorry the arrows didn't show up here. If these are chemi resistors, they can orthogonally keep track of something like concentration. So what we started to simulate are particles that can keep track of time on one axis, that's the x-axis. But on the y-axis, this is a physical axis. The y-axis, they'll keep track of concentration. So the whole device will encode a time-dependent concentration. So imagine a very small cell-sized particle that moves through an environment and keeps track of the chemicals that it's encountering. So it's a very sophisticated function for a very unsophisticated particle. And so here we show, actually, we simulate, how the particle can keep track of time, simultaneously encoding this color is the mem resistor writing. And so as each Y branch powers, it oscillates and powers, and it records the chemical concentration, its magnitude at that time. So here, if you simulate this, if you simulate an arbitrary concentration profile, it will, with some noise, it'll record it. So that's a circuit architecture. You get a very sophisticated function for a very simple fabrication. So I'm interested in that, and we need that in this space. So we have another paper where we've worked out how you can use these probes. You could take probes and have them flow in pipelines. You could look for methane leaks in natural gas. You could put them into plumbing and look for leaks. And then each particle is actually able to record in time its history. And in this work, actually, Ge Zhang is now doing a postdoc at Stanford. He's able to show you're able to map the three-dimensional concentration profile of any arbitrary volume, which is a handful of particles. So actually, you don't need many. I think he showed you can map with some arbitrary precision, just an exceedingly number of particles, in the environment. And so we think this is a vision for some applications in the field. How am I doing on time? Maybe I have a few more minutes. Okay, this I'm excited about. This is actually a collaboration that this is a project that emerged from a MURI that I'm fortunate enough to have on campus with Georgia Tech. My colleagues Dana Randall and Dan Goldman are the theorists and the robotic thinkers that have guided some of this work, and I'm really pleased to have been able to work with them. So this was these are particles like the ones I've shown. Here they're actually sitting at the air-water interface. You're looking down at the air-water interface, and they're in a hydrogen peroxide bath. Okay, and we are they're a polymer disc. They're an SU8 disc, but there's a platinum patch on them. So you saw I have systems like this that I've been showing. There's a lot of work that uses hydrogen peroxide as a motor. So we were surprised when these particles would oscillate. This is actually it's 5 times sped up, but they really do move like this. What's happening is they're forming oxygen from the hydrogen peroxide, making a bubble. The bubble merges and they oscillate. They're being brought together with an attractive force called the Cheerio effect, and also a little bit of the meniscus dome of the. But this beating is actually very, very rhythmic. It's actually it's almost simulating a human heartbeat actually, and it's very regular, and it was a surprising result, and it could be useful. In fact, it's an emergent property that gives us access to a timer. And this is actually the students really wanted you to know. So this is actual experimental data, and the model is here. And actually, there's such great agreement that sometimes people will ask, like theorists will ask, is this simulated data? Whoops, sorry about that. That is not good.
Blank screen button.
Did I do that? Yeah, let me see here. Oh, maybe I can. Don't know if I did this or not. Hopefully, I did not. Oh, okay. Sorry. Okay, yeah, obviously. Okay, sorry. Okay, you can take an FFT of this data. I mean, look this. These particles behave with like precision oscillators. And what's more important is the timing circuit is of the order of what we want to do to control to make a living thing. So it's an electrical engineering secret. It's actually easier to make a megahertz clock than it is a a 1 hertz clock. Actually, you have to frequency half. You have to keep halving it. So the device gets bigger and bigger and bigger to make a slower clock. But this is very regular. You're supposed to see this in this phase portrait. It's called the phase portrait. You're supposed to see because it's a circle. It's a very regular clock frequency. And in fact, what's happening here is when the bubble collapses, it provides a force which gives the particle a velocity. There is a drag. There's a global gravitational force, a buoyancy force that brings the particles together. And there's also this Cheerios effect. It really is called the Cheerios effect. If you've eaten, how many of you have eaten Cheerios? If you'll notice, the Cheerios stick together, and they stick together because of interparticle capillary forces that scientists have named the Cheerio effect. Okay, so it's an adhesion, and we understand it, and it scales, but it brings the particles together while the reaction brings the particles apart. And so there is drag, but you can use these equations to simulate, and the students really want you to know that they wanted to highlight this, that the model in the experiment, this is not simulated data, it's actually real experimental data, and the model fits it pretty well. And we think we know how it can scale. This is just the chemistry of the scale here. And the interesting thing with this work is if you use particles of all the same size, so with 2, you can make an oscillator. With 3, you start to see the oscillation starts to decay. But by the time you're at 5 and 6, and the oscillation becomes chaotic. And so you lose this order. And what we figured out was there's something called an asymmetric an asymmetric ordering. If just one of the particles is slightly bigger, just 20% bigger, it acts as an axle to a hub of a wheel. And so you actually, if you have a slightly bigger particle, you actually resynchronize this whole collective. And it's very counterintuitive. I think nobody would have predicted it, but it gives us, normally for engineers, we think symmetry, precision, you'll get order out of symmetric things. But here you actually had to purposely disorder it. You had to make one not fit, and then you got your frequency locking back. So very counterintuitive, and you can see here you can get the collective actually also to have this regular frequency. You'll actually go from aperiodic to periodic just by dropping in one particle that happens to be a little bit bigger. So I think this is I think when you're looking at these very nonlinear collective emergent effects, I think we need design rules like this that are not intuitive. A lot of our engineering is intuitive, and so we need brilliant theorists like Professor Randall and Daniel Goldman to help to flesh out these rules and help us to design these materials. So that's the story so far. It's not ready for a Disney movie yet, but we're making progress, and I hope and I'd like to thank students, former and also current, and with the hard work. And this work is mostly funded by NSF. We have an Institute for Soldier Nanotechnology, and then also Department of Defense, who also funds this MURI with Georgia Tech. So thank you. Thanks a lot.
Thanks so much for that amazing talk. We have time for a couple questions. I will start. You talked about the range of length scales you were interested in, which I think spanned about 1 micron up to 100 microns. More of the objects you showed were on the 100 micron side.
Yeah.
Is it just harder towards 1 micron? Can you comment on that?
I actually have slides on smaller. It's a good point. The 100 is easier to study for sure. It's also easier to detect, and it's easier to do precision measurements on. But I will. If you go to our papers, we're always careful to show that you can scale down. And so we're doing a lot of our work now at cell size. Although in my defense, a plant cell is about 100 microns. But I'm a
no,
million.
It doesn't mean to be attacking. I was just curious if there was physics or challenges that were emerging for you guys at those smaller length scales that were surprising, or you had to, that kind of thing?
No, you can so all of these things are designed so that they scale down to if your vision of a mammalian cell is 10 microns, then that is our target. Yeah, and so it's a fair point, but it really is it's just easier to characterize at 100 if you're trying to prove a principle and so forth, then it's 100. But if you go into our work, and I showed on graphs, often we were scaling down to 10 microns or 7 microns. But thank you. It's a fair point. Yeah.
Thank you. Looks like your devices or kind of chips are made of mostly SU8-based one. And then is there any effect by the curvature or flatness of the chips in your kind of behavior with robotic devices, something like that?
It's a practical question. We have found that SU8 works very well as a platform. We started a previous MURI with electrical engineering, trying to make cell size devices like this. Some of this work was done. I collaborated with electrical engineers, Tomas Palacios at MIT, and I think they started off by dicing silicon electronics and releasing it into solution. And that does not work. It has a myriad of properties. I can't articulate why it does not work, but many people have tried, and so they eventually moved to the SU8 platform that we have. So is it? I think the question is, does SU8 buckle and change and deform, and wouldn't that kill your electronics? Well, obviously not. It is stable, and I think others have started to use SU8 as a platform. I think because we're grafting nanoelectronic materials like graphene and carbon nanotubes, we actually can tolerate a fair amount of strain and buckling, and that may be unusual for conventional microelectronics. I don't suggest you take the processor for your computer and try to strain and buckle it, or lift it off into solution. I think it's a way of destroying it. So it's but I'm not knowledgeable enough to know why you can't just dice silicon electronics and have them float around. But it's a matter of stability and maybe environmental sensitivity and just maybe a level of precision that you need that you can't get from the physics. Yeah, that makes sense.
Thank you, Michael. You mentioned in the beginning that 2D materials were better for these sensor properties than materials like silicon. I was kind of curious, what was the reason? You said you implied you learned the hard way, but what was the reasoning behind it?
Well, so first, they appear to be easy to graft onto particles. So if I hand you a particle and you wanted to graph electronics onto it, then 2D materials, we know how to do that. We know how to do, you can use assembly. I made these statements. This is the problem with being transdisciplinary, right? I make these sweeping statements and I don't really know silicon electronics all that well. I do know, I've asked colleagues in the field, Paul McEwen, and no one can really articulate why. Maybe you can do conventional silicon electronics and make devices like this, but it's not obvious to me. Your question is there an advantage to 2D materials or carbon nanotubes or nanowires? And I think it's just that. I think with nano, you can actually graft onto an arbitrary surface. You can also get, as I showed, you can get fairly decent electronics for our purposes to work under messy conditions, right? Floating in solution and so forth. And that may be an encapsulation problem for conventional electronic devices, but that's not set in stone. I think the tools we have now reflect the uses they were made for, right? So I'm sure clever Georgia Tech engineers can figure out how to solve that problem, and maybe it's neither silicon, maybe it's gallium arsenide, or maybe it's something. It's a good point.
Okay, let's thank Mike for an excellent talk. Before we head outside for the reception, and I have it on good word that Buzz will be making an appearance. We want to give Mike a token of our appreciation. So what I have here is a framed semiconductor wafer with the Georgia Tech logo etched into it here in our own cleanroom facility in honor of Mike giving the second Oliver Brand lecture.
Okay, yeah.